Here are the results of their experiment. They found that all of the 31-year-old carps were infected with metasaccharida. As for the 2-year-old carps, only 24 out of 30 were infected. Finally, 22 out of 30 3-year-old carps were infected. Next, let's take a look at the metasaccharida itself. Due to the round oval shape, thick outer walls, thin membrane, the presence of ventral suckers, oral suckers, and lack of pseudosuckers, the metasaccharida complied with the prohemostolum's type's characteristics. But physical features can only go so far, so some molecular diagnostics was performed to get more detail. The results of the molecular diagnostics found that 3 Hungarian specimens, Hungarian specimen 1, 5, and 17, were incredibly similar and only 0.4% different genetically, while 6 of the Italian specimens, Italian specimen 1 to 6, were only 0.5% different genetically. But only one of the Italian specimens was identical with Holostephanus dubini, and it was I7, or Italian specimen 7, and it is expected to have a close evolutionary relationship with Hungarian sample 3.
The first time European farm fish were examined for zoonotic flukes was in 2020 by the Bureau of Fish Control. This present study is part of the same program and found 1,000 common carps absent of any flukes. This proved that most European fish farms are free of zoonotic fish farm. The Northwestern farm was found to have A. Spodostoplostomum cuticola metasaccharide. Symptoms of infections may include deformations of fingerlings and black spot appearing due to disease. Infection was found in all age groups with a relatively high prevalence and intensity of infection, but they still can't tell if they belong to either Holostephanus or Cytocotal. To add to this, molecular data is rare and sometimes not available at all.
The reason that the Northwestern farm is infected with fluke may be because it was close to the protected natural area and potential intermediate and final hosts were present. This includes freshwater snail and water birds. Several studies have shown the number of hosts directly affects the abundance and diversity of parasites. As the first intermediate host of cytocolic nematodes in freshwater, the source of infection in Hungarian zoonotic fish may be non-detected infected snail in ponds.
According to the survey result, it can be concluded that Hungarian freshwater fish products are free of zoonotic foods. One class farm was infected with tomatoes, but only with non-zoonotic Cytocotylis species, say parasites unable to infect mammalian model animals, but a species related to this one may infect dogs. The culinary practice and preservation technique of Hungary and most European countries are able to prevent the spread of metasaccharida in the fish fillet, but some traditional practices in Hungary such as smoking fish may not prevent the spread of metasaccharida. Luckily, these practices aren't as common as they used to be.